Welcome to Brain Talk, everyone. Let's talk brains. Dr. Karla Kaun is here to talk to us about what drunk fruit flies can tell us about cravings, addiction, and memory.
Thank you. It's truly a pleasure to be here. I love events like this where the public can ask really cool questions because we get lots of really good ideas from you too. I want to start by telling you a little bit about myself. So I grew up in a remote Métis or Cree community in northern Saskatchewan, and then a remote mining town in northern British Columbia. I went to college at the University of British Columbia, where I completed a degree in biopsychology. And then I went to grad school at the University of Toronto, where I completed my PhD in zoology, a bit of a change from psychology. But during my studies, I really became fascinated with how memories are made and how they can get broken. And so I'll give you an example of this. Neurodegeneration in aging is an example of how memories become more difficult to make, but old memories become really salient and sort of at the forefront of the mind. And addiction is another example of this because in drugs and alcohol are actually a really interesting way to strengthen memories for cues associated with the feeling of being intoxicated. And this can reduce flexibility of memory, and that results in memory staying around for a really, really long period of time. And these are some of the longest lasting memories that we can form. So imagine, for example, the types of memories I'm talking about are ones that you don't think that you're necessarily forming. Maybe it's the dim lighting of a bar. Maybe it's the feel of a glass in your hand. Maybe it's the bouquet of a particular wine. These sensory cues, the smell, the taste, the sounds around you, become associated with the feeling of being intoxicated. And this is true for alcohol and other types of abused substances that affect the brain. So addictions are really unique because they're chronic disorders, and they're characterized by recurring relapses. And these relapses can be triggered by exposure to these cues, like the sound, the taste, the smell, that became associated with the feeling of being intoxicated. And the problem is that we really have no very effective way of preventing these relapses. And part of this is because these cues are all around us, and we don't know really effective ways to block those memories for the sensory experience to trigger cravings. And then the cravings can then potentially induce drug seeking, which can result in relapse. And so we don't yet have effective ways of blocking these types of memories. And around the time I finished my PhD in 2007, I realized that this is a problem that really needed to be understood, but we just don't have the tools yet to tackle this problem at highly mechanistic level in most animal models of addiction. And we certainly can't do it in humans because we don't want to experiment on humans. And so because of this, I joined the lab of Dr. Ulrike Heberlein at UCSF to develop a fruit fly model to study this problem. And you might be thinking, wait, did she just say flies? Yes, flies do form memories. And when you think about this, this kind of makes sense, okay? So in their natural environment, flies like to hang around in orchards, for example, where there's like rotting fruit. And they have to be able to remember where to avoid predators, where to find the best food, and where to find potential mates. And so it's important for them to be able to associate the feelings of good things like food with the place that they were and the cues that were around them. And so alcohol and drugs don't have special dedicated circuits in our brains to induce euphoria, but they do act on our brain's natural reward memory pathways. This is true for humans, and this is also true for fruit flies. So like why flies? When they seem so different than humans, they've been used as a model system for over 100 years. And this means that there's amazing amounts of genetic tools to be able to manipulate almost any neuron in the memory circuit and almost any gene in these neurons in the memory circuit. So we can figure out what are the molecules changing the circuits that and then induce behavioral responses and maybe decisions that move the fly more towards drug seeking. Okay. And so this is sort of an unprecedented level of resolution, and it allows us to understand how alcohol and drugs of abuse influence memory formation at the genetic, molecular, cellular, microcircuit, and neural system levels, all in a behaving animal. So to do this work, we combine a lot of different types of disciplines. So behavior and machine learning and molecular biology and biochemistry and transcriptomics. And because of that, I have a diversity of different types of scientists in my research group. And I find that this really helps innovation. And it's because of that I really welcome and celebrate people from diverse scientific backgrounds, but also cultural backgrounds, because I think their unique perspectives really bring insight and innovation into the work that we do.
Thank you, Karla. This is amazing. So I've known you for a while while we worked together at Brown. And I remember when I first met you, I said, she gets fruit flies drunk. Right? I mean, that's kind of what your research is. And this is pretty novel, right? Because fruit flies have been used in research for a really long time for genetic studies. But the type of behavioral study that you're doing is really quite unique. To drive that point home, I wonder if you could tell people a little bit more about the flies and what other sort of behavioral parallels do you see between the fruit fly and humans, the way they react to the alcohol, et cetera.
Yeah, I mean, it's true. It's the best thing to say at parties, right? Because you're like, what do you do for your job? I'm like, oh, I get flies drunk and high. And we do this through volatilizing alcohol. So this allows us to control the dose. And it's crazy to watch the flies when they become intoxicated, because at first they get a little bit hyper and they start sort of touching each other more. They become more social, if you will, and then they start to become less inhibited. So both male and female flies start to court more, they become more aggressive. And then they can't walk in straight lines anymore.
That's
and
what
eventually they just pass out on the bottom of the vial. What's crazy about this is that about the same dose to induce these types of behaviors in us induces this behavior in flies. And that tells us something about how alcohol can act on any nervous system, something very conserved types of mechanisms. And flies actually do lots of really amazing things. They can learn how to associate shapes together. They have really cool courtship rituals that are really interesting to study. They can navigate incredible places. I mean, they can fly. We can't fly. And so the brain processes required to navigate this three-dimensional space are pretty complicated. And flies are becoming a really increasingly common model to study the brain, because we have a connectome of almost the entire brain. That gives us a roadmap, basically, so we can figure out, like, what are all of the molecular things happening in these types of circuits, because we know how all the neurons are sort of attached to each other in the first place.
Yeah, a map that we don't have for the human brain because it's so incredibly complex.
Yes, in fact, we only have ones for worms and flies at this point, and they're working towards rodents, but it's just such a massive data project. It's going to be a while before we have it as comprehensive.
Yeah. And just to kind of send the message home to the audience, you know, when I talk about the connections that the brain makes, and I always say this to my students, I say, you know, imagine every single device that's on the internet nowadays, every single tablet, phone, computer, server, everything, right? And all the possible connections that can be made. And of course, this is an estimation that was made a couple of years ago, but our brains make more than 5 times the amount of connections that every single device in the world makes online. And that's just in your head. That's amazing. Right?
There are more connections than there are like stars in the Milky Way.
Yeah, right.
That's crazy, right?
Right. So it's amazing to have this model where you have less connections. You can figure out what are the areas, what are they saying to each other.
That's right. It's tractable. So we can put a map together one cell at a time to figure out exactly what's happening when a memory is getting made and when a memory is getting remembered.
And how do you do that? What tools do you use to be able to take a look at the brain of a fly?
So some of the ways that we we do dissect out fly brains, but of course we do this using microscopes, and we use pretty powerful microscopes and fluorescent sensors, like you'll hear from Dr. Abdel-Fattah in a little while, to be able to see how neurons are acting while an animal is behaving. We also use things called optogenetics and thermogenetics, which are basically genes that we've taken from things that glow in the ocean and put into fly brains. And that allows us to shine light on the fly brain, and then the neurons become active. So we can control exactly how neurons become active. And that way we can start to understand, if we make this neuron active, will it start seeking alcohol? And that allows us to really understand which neuron is important for which aspect of the behavior. And the idea is that we really want to understand how to stop the seeking part, or how to stop the craving part, because that's the part about addictions that we haven't quite figured out yet. And the stuff we're doing is very basic science. It's like, when they were studying CRISPR 20, 30 years ago, it was in bacteria and nobody cared, because it was just this scientists kind of putzing away, looking at these special sequences.
Dirty scientists.
They really had no idea that this is going to revolutionize the world and revolutionize medicine, right? And so the kind of science I'm doing is like the putzing away at these sequences and neurons, trying to figure out just how it works, what makes a brain tick. And the idea is that, eventually people will understand that these basic circuit components and actions are really important in driving all types of reward circuits or all types of seeking behaviors. And so what we're discovering now will be able to be applied in more complex brains later on.
So one question that I'm sure people in the audience have is, I think everybody understands that alcohol is bad for your brain, right? At least to some degree, there's an understanding that too much alcohol equal bad. But I was hoping you could expand on that a little bit more. And what does bad mean? Can the alcohol actually change your brain? Can it change the neurons and how they communicate with each other?
Yeah, I would actually go a little farther than that. I would say alcohol is the most dangerous drug in the world. Whoa. And part of that is because it is the most widely used drug in the world, right? And it can affect so many different types of brain cells. And what we're studying in the lab are like these really highly conserved things that are on the membranes of neurons that turn on genes inside of memory circuits. And what we found, for example, is that alcohol can turn on these highly conserved cell signaling pathways. One of them we're studying is called Notch. And our Notch is the same as fly Notch. And in fact, the reason it's called Notch is because 50 something years ago, it was discovered in flies because this mutation caused a notch in the wing. And so it turns on that this notch is also in our neurons and our brain cells, and alcohol can turn it on. And then it turns a whole bunch of genes on and off that are important not only for memory formation, but for cell health and immune response. Mm-hmm. And so, even at moderate levels, alcohol can completely change which genes are turning on and off in your memory circuits. And we think this is one of the ways it's strengthening or changing memories, which sort of enhance the cravings that we have. Because, we get cravings for chocolate, but they're not quite the same as cravings for alcohol or methamphetamine or opiates, right? So what makes it different? We really need to be able to understand these types of things.
Yeah, absolutely. I mean, I think it's just so impactful to think about the fact that, we all have DNA that makes us who we are. It's our blueprint, and that's never going to change. But substances like alcohol and things in our environment can change how that DNA, how your body decides to use that DNA in order to guide your behavior, right? And so that's exactly what you're describing here, is that alcohol is changing how your body decides to use your DNA and what life outcomes you'll have.
Exactly. And it does this not only in our brains, but, in our hearts and in our livers and in our kidneys and most other tissues in our body. So, continued alcohol use can be potentially destructive. Thankfully, humans are pretty resilient and we can handle a lot. But I would say that alcohol is one of the main contributors to other types of factors that, lead to bad health.
Can you speak a little bit on usually when most people take alcohol the very first time, they have a bad reaction to it, but they power through it and they keep taking it, right? So why is that happening? And is there a way we can, we talked about learning a memory, is there a way we can remember that it was bad at the first time, enhance that memory of remembering it was bad at the first time to help us dissuade us from keep going, people that drink a lot to stop?
I've tried to convince people that they don't actually like alcohol, and they're like, no, I love alcohol. I'm like, no, you don't. You actually don't.
I think that's such a fascinating phenomenon, actually. And, it's not just like this in humans. Because the first time you have alcohol, not only it tastes bad, that's why we put so much sugar in our alcohol, right? And like, younger people, or the first time someone drinks alcohol, they're going to drink the sweet wine or the cooler. They're not going to go for like the 40-year-old scotch because it tastes horrible. So there are these negative things. And there's also like the visceral neurotoxic effects that it has. It makes you physically ill, right? But at the same time, and this is what's fascinating about it, at the same time that you're experiencing all of these aversive effects, you're starting to experience the reward effects. And so why, if you went out Friday night and you drank enough alcohol to make you not feel so good Saturday morning, then, a friend calls and asks, oh, we're going out with this other group Saturday night. And you're like, yeah, let's go. So the truth is, we don't understand exactly how that happens yet, but it also happens in flies. And what we do know about how it works in flies is that it seems to simultaneously activate both the aversive and reward memory circuits at the same time. And it turns out, because we have this roadmap in flies, we know exactly how these aversive and reward circuits are interacting. And they interact in really interesting and sophisticated ways. And so the people in my lab right now are actually trying to figure out, there's this one dopamine neuron that goes in and a cholinergic neuron that comes out, and the dopamine neuron encodes the aversive properties and the cholinergic neuron encodes the rewarding properties. And so this is John Hernandez, who's a postdoctoral fellow in my lab. He's trying to figure out, how does this interaction happen? And the idea is that if we can figure that out, then we can look in other types of brains and say, maybe it works like that here, here, and here. And maybe these are the molecules changing. So let's find drugs that target these types of molecules.
Yeah.
Have you looked at, yes.
No, I was just going to say, it's like your love-hate circuits. Love-hate alcohol.
Yes.
Go on,
yes.
Pablo.
Have you looked at the social aspects of addiction in flies or in people? The fact that you can have a terrible experience drinking, but because you're drinking with your buddies, that in itself becomes a reward, even if you are puking at the end of the night.
So, and there's another group that's actually looked at this, and they look at how acutely intoxicating levels will change courtship and aggression. And the aggression that it seems to invoke is male-specific aggression. And they're starting to map sort of the neurons and pathways through which that's happened. We've started looking at it at a slightly longer perspective. And so we give flies 3 doses of alcohol a day for 3 days in a row, and then we take alcohol away. And it turns out when you take them away, flies become crazy social. We don't know exactly why. We think maybe they're trying to seek reward in this social behavior, because social behavior can be a type of reward for flies. But it turns out that this is highly correlated to massive changes in the immune response genes. And so we actually think that this social behavior is somehow correlated with changes in immunity inside of the brain. And maybe, social behavior in other types of organisms is also correlated with this. And at Society for Neuroscience this coming fall, there's going to be a symposium about this in rodents, actually. So I think it's very likely that these types of behaviors are correlated. We just don't understand how or why yet.
Pretty exciting. To piggyback on that, is there a difference in how much they drink if you put a fly by itself versus if you put them with their buddies?
I was going to ask that question too.
Because now we're getting to COVID, right? Like us drinking by ourselves at home in our pajamas.
So we haven't, socially isolated flies for long periods of time and compared them with group housed flies to test if it changes things. But we do know that these types of social behaviors can affect it. For example, male flies that are sexually rejected 3 times a day for 5 days in a row double their alcohol intake. And it's because they find alcohol more rewarding. And so I think it's very likely that these social behaviors can modulate that. And we know this happens through neuropeptide Y-like signaling, which we share with flies. In flies, it's called neuropeptide F because it's slightly different, but it's very highly conserved in how it acts. And so I have no doubt that there are influences like this. We do know within a group of wild type flies or like control flies. We have some flies that like to increase their alcohol self-administration over 3 days, and then some flies that like to decrease it. But if you increase the dose of alcohol, you get more and more flies that will increase their alcohol self-administration. So there's a lot of variability in fly behavior, and we just sort of are starting to explore this type of variability now.
And that's similar to humans, right? I mean, some humans will drink in college extensively and then move on and grow up and not continue to abuse alcohol, whereas some people will continue to abuse alcohol. Some people are more predisposed to it. So can you tell us a little bit more about, what does that predisposition look like? Do we know what makes one person more predisposed than another?
So I say that there are a lot of scientists around the world investigating these types of things. And I think that one of the predictors of whether someone is likely to develop an alcohol use disorder is, for example, age at first alcohol intake. And another is early life stress. And so we haven't started investigating this in flies yet, but I think there's a lot of data and other types of models to suggest that early life stress certainly can change that thing. What really fascinated us about our data is that only about 30%ish of the flies would increase their self-administration. They would start to escalate. And in humans, only about 30-ish% of people will develop an alcohol use disorder. And so we don't yet know what the underlying mechanisms for that are. But the cool thing is now we have an opportunity to study it at a very detailed level.
Yeah. Is there a genetic component? Do we have an alcohol gene? Have we found it yet?
Well, alcohol use disorder and sort of alcohol behaviors are very complex behaviors. And so I can tell you there isn't just one gene. It's really complex. It's polygenic, meaning many genes. And these many genes are probably interacting in really interesting ways. And it's not always the same set of many genes per person. And so it's been really traditionally very, very difficult to narrow this down, especially in humans where you don't want to manipulate genes. That's partly why we study it in flies, is that we can study it one gene at a time to understand the contributions of each of these genes, to understand things at a mechanistic level, or even 2 or 3 genes at a time, to slowly start putting that gigantic jigsaw puzzle together.
Right. And you can isolate the genetic components from the environmental ones, right? Because presumably having a parent who was addicted to alcohol makes you more predisposed. But is that because of a sort of social imitation factor, or is it because of the genetic factor? Is it, combination of both is more likely, but you're able to do that in flies?
Absolutely. Yeah, we have very rigorous environmental and sort of like learning type controls that we do. And we can make sure that the flies are all little genetic clones of each other. So we can control these extremely rigorously.
Is there anything unique about those 30% of flies that do ramp up their alcohol use? Have you done any correlation where you can look at behaviors after you, isolate those 30 or before and then see which ones fall in that 30% category?
I would say that is, I wish that my postdoctoral fellow John is watching this, because that's basically his career in the making. That's an excellent question, and we don't understand yet now, but like I said, we have a great opportunity to really dive into the mechanism through. We can look at genetics, we can look at what genes are changing rather than just what genes are different. And then we can look at, how each of that contributes to neural dynamics and how that then influences behavior. So one of the experiments he's planning next is, can we activate one of these neurons to induce escalation or get rid of escalation of alcohol use or alcohol self-administration?
I'm having difficulty removing from my brain the thought of a fly sitting on a bar with a cigarette. But have you conducted any studies using cigarettes instead of alcohol?
So we actually have in collaboration with Norma Velasquez-Wola, who's at Lewis and Clark College in Oregon. So she studies the effects of nicotine in early development, and together we're starting to develop a model to see if nicotine is also rewarding to flies. Their memory circuit is highly cholinergic, has tons of these receptors in it. So we think this is very likely. But she's definitely shown that in development, like alcohol and nicotine exposure interact in really interesting ways, much like they do in rodents and in humans.
See, now you have the same image in your brain.
Yeah, exactly. And you need to use that and make a lab meme out of it. You need to get one of your students who's really good at art, and that needs to be your logo for your lab. All right, we have about 1 more minute left. And so I want to leave it with, you mentioned treatment in one of the answers that you were giving. And I know that there are some treatments out there aside from therapy treatments, right, where you're kind of working towards getting better, but there are also some pharmacological treatments, right? There are some that are aversive, that make you have a bad reaction like you did the first time. There are others that that block your receptors for opiates, correct? So can you say something about those treatments, and maybe, you know, what kind of treatments your lab is hoping to add to that toolbox in the future?
So there's lots of different treatments out there. And I just want to say for anybody listening that it's a combination between the pharmacological and behavior therapies that have really been shown to be effective. And there really are effective treatments out there for anybody that's interested or looking. Because, you know, all of us are in some way or another impacted by use disorders. I would say that at this point, the pharmacological treatments alone aren't always the most effective. And that's because people don't necessarily want to take them. Like antabuse, that thing that you take when you're drinking and it makes you viscerally ill and want to throw up. That's not very fun. And it can affect your life in ways that you maybe don't expect. And so we want to be able to make treatments that are a little bit more effective, not in acutely blocking the effects of alcohol or opiates or something like that, but in reducing the cravings. And so we think the way to do this is to really understand what are the basic mechanisms that are mediating these sensory memories for the intoxication experience. Because we think that alcohol and drugs are affecting the molecular mechanisms in these circuits, and that's what's making the memories somehow stronger or different. And so if we can sort of, you know, pick apart what are the mechanisms that are changing, then we can potentially find drugs that are targeting exactly that mechanism. Right. And by reducing cravings, maybe we can reduce relapse to make life a lot easier. So I think in the future, it's going to be some combination of lots of these different types of treatments, you know, the acute the pharmacological and behavioral treatments, and then later on, once people's lives are stabilized, maybe we can have these types of drugs that are helpful for reducing cravings. I don't know. Only time will tell.
Only time will tell. And I know that we're at time now, but you know, it's so important to realize the difference between craving and pleasure. For people who are very far down the line in terms of their addiction, they end up not actually enjoying the alcohol or the drug of abuse or the opiate. They don't take pleasure in it, but they need it, they crave it. So absolutely looking at the craving, you know, why do we crave and what drugs can we develop in order to stop those cravings is really important. Thank you so much, Karla, for being with us. I hope you have a great rest of your day and happy Brain Week.